So the topic of I'm Manvendra Patel, and the topic of my presentation is biochars and engineered biochars for aqueous pharmaceutical removal. A brief overview in this presentation. First, I'll be giving a brief overview upon what biochars are and how they can be engineered, and then I'll provide a brief overview about the pharmaceuticals as contaminant. Then we'll move on to our presentation regarding biochar development, their characterization, and sorption studies. So, biochar are solid materials obtained from the carbonization of biomass. Biochar can be developed through slow pyrolysis, fast pyrolysis, and gasification. Although people are using microwave pyrolysis and hydrothermal pyrolysis also nowadays. The biochars can be utilized in a large number of problems. For example, wastewater treatment, soil amelioration, anaerobic digestion, building materials, agriculture, gas storage, and all these problems make biochar a sustainable solution for the environmental problems remediation. Engineered biochar production techniques. As we all know, biochars can be produced through a large number of biomasses: woody biomass, agricultural biomass, forestry, animal residues, industrial waste materials, and through different techniques of pyrolysis, the biochars can be prepared. These biochars can be modified or engineered through three main techniques: biological activation, biological methods, physical methods, and chemical methods. Biological methods utilize the digestion and using of microbes. Physical activation uses gas, steam, microwave, ball milling, and magnetization. In chemical oxidation requires chemicals such as hydrogen peroxide, alkali, acid, and their coating or impregnation. These techniques provide desired properties to the biochar as required for a particular process. In our study, we utilize the pristine biochars and we magnetize the biochars using co-precipitation method. So the question arises, why did I choose pharmaceuticals for my study? As we all know, pharmaceuticals have arisen as the contaminants of emerging concern in the past decade. The pharmaceuticals have been reported in the world, and more than 600 different kinds of pharmaceuticals. So pharmaceuticals is just not a single problem. It is a group of large number of contaminants which individually and together can create a problem which requires immediate attention. Problems related to pharmaceuticals are they can be pseudopersistent, they can bioaccumulate, they can induce toxic effects on flora and fauna individually, and also when in natural environment when pharmaceuticals are present in a complex group or a complex mixture, they can impart a variety or different kind of toxic effects on this. Present treatment techniques including wastewater and sewage treatment plants are inefficient and requires advanced treatment methods such as AOPs, adsorption, and catalysis. Although AOPs, catalysis, reverse osmosis are widely used in developed countries, but when it comes to the developing nations, adsorption can be a pretty good option, because if you are using low-cost adsorbents developed from locally available materials, adsorption can provide a pretty good method for the removal of these emerging contaminants. The application in less technical or skilled personnel requirements. So for the study, we have chosen two different groups. One is antibiotic and another is analgesic, because these groups are some of the largest used groups and largest found groups in environmentally relevant waters. So as we can see that antibiotics antibiotics are most used, one of the most detected compounds. Thus, we have used a representative of antibiotics, ciprofloxacin. In another, we can see analgesics can be seen all over the world in surface waters or different wastewaters. Thus, we have or commonly called as acetaminophen as a representative compound for the study. Development of biochars. For the study, we have developed a series of biochars using water hyacinth and banana peel. Both these, water hyacinth is a very big invasive problem. Thus, it is used, and banana peel is largely used in household and municipal crops. Thus, two different biomasses from two different problems are taken and developed into a series of biochars from 350 to 750 degrees Celsius. These biochars are prepared, grinded, washed, and further used for adsorption studies. For the engineering of biochar is converted using magnetization method through co-precipitation of ferrous sulfate and ferric chloride in biochar and mixed with co-precipitated with aqueous and AOH and in the presence of heat. Then for 24 hours with aging of 24 hours and filtered, and the magnetic biochars are filtered and washed. This method is taken from Mohan et al. 2021. Characterization. The developed biochars are characterized for a variety of physicochemical and morphological properties such as SAM, TAM. SAM and TAM are used for the surface morphological evaluation of the biochar. FTIR for functional groups for estimating the various functional oxygen containing and other functional groups on the surface of the biochar. Elemental composition, surface area measurement through PET surface area will determine and evaluate the surface pore and porous surface area and pore structure. The magnetic moment of the magnetic biochars has been evaluated. XRD for mineralogical properties and the elemental composition using CHNS, XRF and ICP for bulk element composition and SAM-ADX for the surface element composition of the biochar. So now we are seeing the SAM images of banana peel biochar, which shows a large number of porosity ranging from micro to mesopores. The selection of these biomass is basically based upon two important problems. First, they are available in the plenty in local environment, and second, after drying, these biomasses provide a porous material in itself, which is also visible after their conversion into biochar, which shows a large number of pores and honeycomb-like structures, canal-like structures, honeycomb-like structures, and these pores provide extensive surface area up to 600 meters square per gram in case of banana peel biochar 750, which provides extensive area for pharmaceutical adsorption. As we all know that organic contaminants require large surface area, so that is also the main reason behind the selection of biomass. So surface elemental composition of the both the banana peel and water hyacinth biochars is shown in this slide, and which shows that both biochars are rich in carbon and oxygen, although the presence of other elements such as calcium, potassium, phosphorus are also visible, which shows that the SAM image of engineered magnetic banana peel biochar. As we can see, at low magnification, the SAM images does not impart any changes, but as the magnification is increased, both the spherical iron oxide shapes and the spindle-shaped iron oxide clusters are easily visible on the surfaces of the biochar, which shows the presence of iron oxide precipitated in the form of uh, ferrous and ferric oxides, FTIR spectra. FTIR spectra was performed and it shows the plenty of functional groups in the region of hydroxyl and carbonyl groups, hydroxyl, carbonyl, carboxyl, and these groups are in plenty, both aromatic and aliphatic groups such as CH bending and stretching were also visible. And these groups are further responsible for the sorption and complexation interactions between the biochar and the pharmaceutical compound. Sorption studies. Sorption studies are performed in two modes, both batch mode and continuous mode to column studies. The batch mode were used to optimize pH, kinetic effect of dose, effect of concentration, effect of particle size to kinetic studies, and isotherm studies were performed to optimize and evaluate the impact of initial solution temperature upon the sorption process. With these studies, the effect of pyrolysis temperature on the sorption process, effect of particle size on the rate of sorption, and effect of different multi-components such as ionic strength, cations, and anions will be evaluated and further estimated. Correlations. These studies provide various correlations between biochar properties with the pyrolysis temperature and pharmaceutical sorption, where we can see high correlations between percent oxygen, H/C ratio, O/C ratio, percent carbon, CY/N ratios, and O plus NY/C ratios, and S content shows high correlation with the sorption of acetaminophen and ciprofloxacin. Although ciprofloxacin showed higher correlation as compared to acetaminophen in case of percent oxygen and percent carbon, and acetaminophen shows higher correlation with percent S content of the biochar. Kinetic studies showed that increase in particle size increases the sorption kinetics. However, the isotherm studies of acetaminophen showed with increase in pyrolysis, with increase in temperature, the sorption increases. However, the sorption of ciprofloxacin does not increase with increase in sorption temperature. In case of magnetic biochars, the sorption temperature, with increase in sorption temperature, the sorption of both acetaminophen and ciprofloxacin increases in case of both banana peel and magnetic water ion biochar. However, the sorption capacity increases rapidly as compared to pristine biochar, where we can see the ciprofloxacin sorption capacity of around 20 milligram and around 55 milligram, maximum 55 milligram per gram is achieved for pristine biochar. However, the capacity increase up to 180 milligram per gram for acetaminophen and up to 40 milligram per gram in case of magnetic biochar, which shows the importance and role of the development of iron oxides and minerals present on the surface of biochar. Sorption mechanisms of biochar for the pharmaceutical removal shows the complexation mechanisms such as electrostatic attractions, electron donor acceptor, surface complexations, hydrogen bonding, cation bridging, and charge assisted hydrogen bonding. Along with these, pore diffusion also plays major sorption interaction with in case of pharmaceuticals. conclusions of this study is that both banana peel and water hyacinth was a potential candidate for pharmaceutical sorption and provide the efficient sorption capacity. Although magnetization process increases the sorption capacity rapidly, in case of acetaminophen, maximum 55 to 180 milligram per gram, and in case of ciprofloxacin, 20 to 40 milligram per gram. And the role of hydrogen bonding, van der Waals forces, and pi-pi interactions are important in case of explaining the sorption mechanism. And now we are ready for the questions.
Thank you very much, Manvendra, for your presentation. And I think the sound quality was good enough, so we could hear you well. So you got a question from Frank Brigano. He's asking you what method did you use to measure the surface area? And if you used the BET method, which gas did you use for that?
We use the BET method and we use nitrogen for that, and to adsorption, desorption isotope.
And I also have a question, because you said that the magnetization increased the sorption capacity. So how did the magnetization influence the biochar yield? Did you get a higher yield or lower yield, or how did that change?
We did not do the pre-treatment of the biomass. We developed the biochar first, and the developed biochar is magnetized in the later stage. So we does this in a post-treatment method. So it is
okay.
So the yield estimation is completely different now.
You got another question from Paul Anderson. Do you expect other types of source biomass to give similar results? And which ones would you expect and why?
Although during my studies, I have tested various biomasses such as wheat husk, rice husk, and corn straw, but these biochars didn't give the desired results, including sugarcane bagasse also didn't give the desired results. I think some of the porous biomasses such as algae, biochars derived from the dried algae, and these can provide porous surface area, which can provide similar or better results.
Okay,
or some other invasive porous invasive species such as water lily or these can provide some high surface area.
Did you try other feedstocks than banana peel and water hyacinth before?
Not before. We have used a large number of biochars for the sorption trials, but before this study, we didn't use water hyacinth or banana peel.
Okay, we got a question from Felipe Rego as the next one. So were the higher temperature chars better than lower temperature chars?
In sorption of these pharmaceuticals, these chars doesn't work better because they doesn't provide enough sorption capacity. The results are not shown here, but these results are going to be published soon and in the communication process in a journal. So they will be available soon online. So which shows that these lower temperature biochars are much less inefficient in providing the sorption of these pharmaceuticals, ciprofloxacin and acetaminophen, as compared to the sorption capacity at 750 and 650.
Okay. And then we have two last questions, one from Praveen Jagdal.
Yes, Jagdal.
Yeah.
Thank you very much. Hello, Manvendra, Professor Manvendra. Nice presentation. Nice.
Thank you. Thank you.
I have just one question, because you are talking about the banana peels. Okay.
Okay.
So basically, first thing is they have water content. So if you're drying them before pyrolyzing or you're directly pyrolyzing them, this is my first question. And also, this is a waste byproduct, so it can be used by animals for the food. Okay. So how much you are
Exactly.
thinking it can be commercialized for the in the future? Thanks.
Thank you. So first, the first answer of your question is that we are not using the wet biomass for the biochar preparation because biochar preparation through slow pyrolysis or fast pyrolysis method requires dry biomass. So we are sun drying the biomass because in India there is a lot of sun and lot of heat, so we are using directly sun drying to not increase the cost. Okay. And sun drying is providing the desired results. And the second question is, pardon, can you explain the second question again?
Related with the commercialization of this
Okay,
process.
okay, okay. So India is the largest producer of banana peel. And India is also the largest consumer of banana peel around the world, with almost 70 million tons of bananas are produced every year. So, and banana peel consists of a large content of the total banana biomass. And in everyday usage and along with industries and places, bananas are used in a large scale. Thus, utilizing banana solution, although animals can feed on banana peel, but when you are talking about the industrial scale and household scale, and you are just people are just throwing banana. For example, in our mess, I'm living in a university campus, and university campuses contains large masses, and where a large number of bananas are used every week, and all of it is thrown to the garbage, and which ultimately ends up in landfill. So utilizing these kind of waste can also provide a solution for municipal waste generation.
Okay, thank you very much, Manvendra. If there are any more questions, so you got some more questions in the chat. I ask everyone, please write your question in the chat, and you can.
I have a question, please, if I can ask.
Okay, but it's the last question now. So
yeah, I will ask a similar question. You know, say if you want to use banana peel as your feed, so you have to put like the waste bins that only connect that can only collect banana peel, nothing else. This seems to be practically very, very difficult, or you have to sequence the waste and separate the banana peel. That's my first question. And the second thing is that when you talk about pharmaceuticals in water, there may be hundreds of pharmaceuticals. So how will you imagine how much is it efficient on one or how much is it efficient on the other one? Thank you.
Okay, so answer of your first question is that truly it is waste segregation is always a problem. But when we talk about institutional waste collection, for example, in industries, fruit juice or fruit, these kind of industries where a large scale of bananas are used every day. Those industries, from these industries, you can collect a large scale of banana, which can be utilized for the commercial purposes. And in other institutions, where just like bakeries and all those things, and collection from these kind of institutions can provide a sustainable solution for this kind of idea. For your second question, pardon, can you explain the second question again?
Yeah. My second question is that in the water, there may be numerous pharmaceuticals, not one, not two, not three.
Okay.
So many. So how will you assess how much is it effective on certain pharmas?
This is what I have presented is just the first part of my study, and currently I am in the process of working in the process of utilizing these materials for a large number of pharmaceuticals, and we are preparing some filtration kind of units which can be utilized for utilizing these biochars for pharmaceutical remediation. So currently this study is under process and we are working upon it.
Well, thank you very much, Manvendra.